Hello, dear all, dear colleagues. Welcome to my presentation about epigenetics and fetal programming. First of all, I would like to thank the organizers of MDPI and GENES for sharing this fantastic conference with us, which will take some days now in November of this year. I'm going to talk about fundamental features of epigenetics first, and will then try to deduce which implications that may have for animal breeding. And in the second part of my talk, I will focus on the phenomenon of fetal programming, which has especially implications in animal husbandry and management. I will start sharing my screen with you now. So my name is Klaus Wimmers. I'm working at the Leibniz Institute for Farm Animal Biology, called FBN, in Dummersdorf, which is close to the Baltic Sea in Germany. And the title of my talk is Epigenetics and Fetal Programming, New Opportunities in Animal Breeding. First of all, I would like to ask you to think about the fact that a zygote, a single cell with a certain genetic information, becomes a complex organism which is about 10 by the power of 14 cells, all containing the same genetic information, but of course they are very diverse in terms of structure and function. So there's obviously a second level of information in addition to the DNA, the genome. The genetic information contains the general instruction for the manufacture of proteins and RNAs. It's the DNA sequence. It is the potential, what could happen overall. And the second level is represented by epigenetic informations, which contain the instruction on how and when and where these information should be used. And this is mediated by epigenetic marks. In fact, epigenetics therefore refers to ontogenetics and the cell fate determination. And this is where the term epigenetic comes from. The use of the term epigenetics and its definition has changed throughout history. Conant-Weddington in 1942 used this term for the first time, and he stated that epigenesis describes how a genotype leads to the formation of a phenotype in development. Later on, Robin Halliday mentioned that epigenetics is about the temporal and spatial control of gene activity during development of complex traits. A current definition states that epigenetics is the study of changes in gene functions that are hereditary to the progeny of either cells or organisms, so it is mitotically or meiotically heritable and that occur without any changes of the DNA sequences. Epigenetics is used in medicine, for example, and serves there as a clinical biomarker for diseases. Epigenetic is, for example, used to support diagnosis and prognosis in cancer and to predict a patient's response to particular treatment. In animal breeding, we are interested not only in predicting the phenotype of an individual, but also in predicting its potential to inherit trait. The fundamental statement in animal breeding is that environment and genotype make up the phenotype. And we are interested in analyzing the relationship between the genome and the phenome. And nowadays, we have a lot of especially holistic approaches that enable us to uncover this black box of the connection between genome and phenome. And thus, nowadays, we analyze the relationship between the genome, the transcriptome, proteome, metabolome, up to the organismal phenome. And there is increasing evidence now that epigenetic variation contributes to phenotypic variation. So knowing it could improve the prediction of the phenotype. And we also learn that epigenetics provides a link of environment to the genome. So epigenetics is involved in genotype by environment interactions. So epigenetics is about molecular features of DNA and also RNA, others than nucleotide sequence variation that regulate gene expression. Important features of epigenetics are DNA nucleotide modifications, DNA methylation, which usually takes place at the cytosine sites in neighborhood of guanine site, and we mention this, the CpG sites. So CpG methylation is one of the most important and best analyzed epigenetic marks. Other epigenetic mechanisms include histone modifications. Histone modifications can be methylation, acetylation, phosphorylations, and others that occur at specific amino acids of different histone proteins. Also, RNA nucleotide modifications, RNA epigenetics is established now, and sometimes also non-coding RNAs such as microRNAs or long non-coding RNAs are also treated as epigenetic factors in the broadest sense. I will focus on DNA methylation today. And as already mentioned, DNA methylation takes place at cytosine residuals, usually in connection with guanine. DNA methyltransferases catalyze the methylation, and the DNA methyltransferase 1 is involved in the maintenance of DNA methylation, which is necessary after replication to get both DNA strands on the same methylation status. Other DNA methylases are involved in de novo DNA methylations. These are the DNA methylases 3A and B, and they need a number of cofactors like Phi RNAs and some sequence motifs that are involved in these processes. Epigenetic markers can contribute to phenotypic variation and are set already early during embryonic development and lead to different methylation patterns in individuals and populations. DNA methylation of the pronucleole in the zygote occurs immediately after fertilization. Afterwards, the establishment of methylation, which is mediated by DNA methyltransferases, begins already in early embryonic phases. Genetic factors influence the pattern of epigenetic markers, but also the intrauterine environment can affect the establishment of DNA methyl pattern. Overall, the epigenome is shaped throughout the whole postnatal life. There are stable modifications of the epigenome that are inherited methodically, but there is also lifelong remodeling. Epigenetic modifications, even acquired modifications, are also inherited meiotically. That means that there is also transgenerational inheritance of environmental epigenetic modifications. Thus, epigenetics mediates effect of ancestral, past and present environmental factors. With regard to the implication for animal breeding and husbandry, the causal role of epigenetics for phenotypic variation is important, and there are three aspects that are of interest. On the one hand, the methylation status of a locus can be driven by nearby genetic variation, and differential DNA methylation then affects the phenotypes. So this is the occurrence of so-called methylation QTLs. On the other hand, genetics variants can be modified by their neighboring epigenetic marks, so that the expression of a particular variant depends upon the methylation state of a particular allele. Finally, I also would like to mention that methylcytosine is a hotspot of mutation. Methylcytosine can be changed to thymine by desamination, which is not detected by repair mechanisms of the cells and leads then finally to a CT transition, which has much implications also for the evolution of all organisms. I would like to explain a little bit about the potential causal role of epigenetic marks. And here on this transparency, we have the situation that the SNP determines the methylation. So we call this a methylation QTL. In the homozygote carrier of the A allele, the neighboring cytogene is methylated. The gene is not expressed, and consequently we have a phenotype X. The alternative allele T leads to methylation leads not to methylation of the cytosine. So in this case, with the allele T, we have an expression of the respective protein, and this leads to the phenotype Y. So here in this situation where we have methyl QTLs, the SNP and the DNA methylation site are always in linkage disequilibrium. That means in a genome-wide association study, we would identify this SNP, and we may use this SNP also in breeding. The causal role of the DNA methylation here in the expression of the trait will only be identified if we are looking for DNA methylation in this case. Here we have another situation. Here we have the situation that the two alleles A and T of a polymorphic sites occur in combination with a methylated CpG once and with an unmethylated CpG on the other allele. In the heterozygous individuals, we observe different phenotypes. Methylation therefore influences the effect of the SNP here. The SNP and the methylation occur independent from each other and are not in linkage disequilibrium. In a genome-wide association study, in this case, we would not detect this SNP. And of course, we would also not identify the role of DNA methylation here as a cause involved in the expression or non-expression of a certain allele. So the origin of DNA methylation can be genetic, so stable, inherited, and leading to individual and population-specific methylation patterns. And on the other hand, DNA methylation can also be generated by the exposure to environmental factors. The consequences of DNA methylation depend on the genes that are involved, whether it is a gene for a quantitative trait loci, a gene for hereditary disease, a gene for the response to stress, or other environmental factors. DNA methylation can be a cause of the phenotype, as we have seen. It is involved in genotype-environment interaction, and especially this is the case if genes are involved that are responsible for response or responsiveness to environmental factors. Moreover, the epigenetic markers can of course serve as biomarkers to predict the phenotype. So, epigenetic marks can be stable. That means they are transferred to offspring and are likely to be in linkage disequilibrium with an SNP, independent of the fact whether it is a methylation QTL or not. So in this case, they do not add much to uncover missing heritability, meaning the inherited causes of variation of complex traits that have not been yet identified. However, the epigenetics marks may be causal, and knowing the causal of the establishment of a certain phenotype, especially in complex traits, is very important to promote breeding. If epigenetics marks are unstable, wiped clean in the early embryonic development, then of course they are not a good selection criterion. They do not contribute to the missing heritability problem, but they still may be biomarkers for phenotypic prediction. So this is not all just theory. In fact, there's a growing evidence that epigenetic marks are heritable. I like to refer here to the so-called POP CPGs that were identified in humans and that show differences between American, Caucasian, and Chinese Americans. There are more than 400 of these so-called POP CPGs known. The heritability of DNA methylation has been estimated to be about 0.2. And it has been shown that 20 to 28% of individual DNA methylation differences are due to sequence variation. So they are involved in the situation of the existence of methylation QTLs. And in fact, overlapping has been detected between methylation QTLs and so-called eQTLs, expression QTLs, that reveal the effect of a single SNP on both DNA methylation and expression. Interestingly, methylation QTLs were enriched in metabolic traits or in disease loci. So this collection of facts made us to look for DNA methylation pattern in farm animals. And I would like to start to mention here an example of a study conducted in chicken. Beltecke and his colleagues selected red general fowl, which are considered as the ancestor of our current farmed animals, for high or low fear to humans. And after only 5 generations of selection, they found differences in DNA methylation profiles of the birds. As you can see in this Manhattan plot, in which each dot represents a window containing CpG sites assigned to their position on the chromosomes along the x-axis, that show significant windows, which is indicated with the p-values on the y-locus. The colleagues analyzed the hypothalamus of the animals and detected methylated DNA sites by immunoprecipitation sequencing. And they scanned the whole genome in 1 kb windows. They found nearly 1 million windows of 1 kb size that contain about 10 million CpGs, and about half of it were higher in one selected line, and the other half of it was higher methylated in the other selected group. Overall, there were 22 significant windows as an FDR of 0.1, and these 22 windows contained 187 CpGs that were differentially methylated between those animals with high and low fear to human. Interestingly, the colleagues found no overlaps between the differentially methylated sites and the differentially expressed regions that they have detected before. But interestingly, some genes that were differentially methylated were in regions that are associated with neuronal functions. So, in summary, this study provides a clear evidence that even after short-term selection of only 5 generations, we observe divergent epigenetic patterns. So we have a strong impact of selection on DNA methylation, and this is inherited from generation to generation in this selection scheme. We studied differences of DNA methylation patterns between breeds of pigs and at the background of distinct genotypes of a major gene, namely the ryanodine-1 receptor, that affects the intercellular calcium flux and has large effect on the muscle and fat metabolism. We compared muscle DNA methylation in animals of the breeds Duroc and Biatrin, which differ largely in fat and muscle metabolism, and also at the background of the 2 genotypes at the ryanodine receptor 1, and we also addressed the F2 cross of the 2 breeds Duroc and Biatrin. We found about 400,000 CpGs by reduced representation Bezel-fit sequencing that were mainly located in promoter regions, intergenic regions, and introns. And we only found a few CpGs here in the red area in coding sequences, which is expected. Hierarchical clustering analysis of individual samples of all 440,000 CpGs of the 4 groups revealed a nice clustering according to breed and genetic group. Manhattan blocks showed here differential methylated CpG type between the breeds and that were identified all over the chromosomes. And in this volcano plot, you can see that we selected or identified significant CpGs with regard to the p-values and with regard to the differences in DNA methylation, which was above 25%. So in fact, at an FDR of 5% and with at least 25% differences in methylation, we detected overall some 4,600 differentially methylated CpGs. More than 2,000 were differentially methylated between the pure breeds, and about 1,300 were differentially expressed between the pure breeds and the crossbreeds. Interestingly, between the two groups within the Pietin breed, differing in the ryanodine 1 receptor genotype, we found 1,060 differentially methylated sites. So of course, these differentially methylated sites are close to genes, and we did a functional annotation analysis of these genes and looked for enrichment of genes in certain canonical pathways or co-terms. And I just want to mention here a few interesting pathways. So we found differences in the thyroid receptor and retinol X receptor activation of Duroc against all other breeds, which is interesting because this pathway is, of course, involved in a number of biological processes such as growth, development, and metabolism. For the comparison between Duroc and Pietin, we also found functional enrichment in glucose metabolism and Wnt calcium signaling, also pathways related to metabolism and growth. And most interestingly, between the two ryanodine 1 receptor genotype groups within the Pietin group, we found an enrichment of genes related to calcium ion binding, so which is an indication that there are probably some compensatory mechanisms active between these two groups differing in calcium metabolism. The integrating of the crossbreeds in this analysis gave us the opportunity to look for those CpGs which are of potential Duroc or Pietin origin. So what we did is we selected first those CpGs that showed no differential methylation between Dupi, the crossbreed, and Duroc, but that differed between the crossbreed and Pietin. These were 408 CpGs that are likely of Duroc origin. Vice versa, we looked at the CpGs of Pietin origin by selecting those with no differential methylation between Dupi and Pietin, but with differences between the crossbreed and Duroc. And here we found 804 CpGs of Pietin origin. We selected a number of genes for exemplarily validate the association between differential methylation and differential expression measured by real-time PCR. Here's one example. The 6 gene shows high expression in Duroc and low methylation in Duroc compared to the Pietin breeds. This gene has regulatory functions in lipid metabolism, so also as an indication that for the differences in lipid traits between the two breeds. And another example is here between the both ryanodine 1 receptor genotypes within the Pietin breed. Here we found genes that are related to the calcium functions. For example, here the EHD3 gene that shows lower expression in the NN genotype, but higher methylation compared to the PP genotype. So calcium, the EHD3 gene acts and calcium channel function as a key regulator in the ion trafficking, and again, this may represent a compensatory effect in response to the ryanodine receptor mutation. I would like to mention another last example. We looked for animals that differed in their coping behavior, and we selected so-called pro and reactive animals. And in a previous genome-wide association analysis, we identified SNP haplotypes on chromosome 12. And here in this study, we used animals with the typical SNP haplotypes and the typical phenotypic expression of pro and reactive coping behavior to look for differential expression in some brain areas, and then we also looked for differential methylation. Both differential expression and differential methylation allow discriminating the two groups in heat maps, and interestingly here in the Manhattan plot you can see that we detected a number of differentially methylated sites on chromosome 12 in the region of this QTL. And just to view in one example for one gene, you see that within one gene we find several CpG sites that show these contrasts between the proactive and the high reactive animals. So to summarize what we have shown here is that we found that DNA methylation profiles are the result of selection processes. We find this in long-term selection, so as differences between breeds. But a study in chicken has also shown that short-time selections shape the DNA methylation profiles. And we found that also at the background of different genotypes and haplotypes of myogenes and related to different behavior, for example, there are differences in the methylation profile. So there is clear evidence for a stable inheritance of methylation profiles over generations. And this makes also clear that DNA methylation really contributes to phenotypic variation, and it adds to clarifying the heritability of complex traits, and especially it contributes to our understanding of causal mechanisms of traits. But the DNA methylation pattern are still of limited use for selection, because if they are stable, as mentioned, they are in linkage disequilibrium with SNPs that are easier to detect. However, they are also useful and still useful as biomarkers for the prediction of individual phenotypes and also for the response to environmental factors. And that brings me to the second main topic, the fetal programming. So fetal programming or nutritional programming is a phenomenon that was found from epidemiological studies that shows that offspring of mothers that had bad dietary situation during their pregnancy do suffer from metabolic diseases more often. From this, Hales and Barker derived the fetal origins hypothesis of adult metabolic disorder. This hypothesis says that non-adequate fetal nutritional supply leads to adaptive processes which have obvious immediate effects as low birth-wise and disproportional organ weights, but this adaptation also covers nutritional programming. That means a predisposition for metabolic diseases like diabetes, hypertension, and obesity in adult life. So there is a long-term effect, and additional effects of early postnatal diets may adhere. This nutritional programming leads to the thrifty phenotype, and this thrifty phenotype hypothesis says that individuals surviving adverse interroutine nutritional conditions are prone to adult disease when exposed to adequate or excessive nutrient and energy supply. So if this, of course, seen from the human medicine side, is right, then the question is whether we could probably use these 30 phenotypes to produce animals that are more effective. So nutritional programming means that maternal diets during gestation can lead to programming of the genome of the offspring in utero and lead to alteration of the offspring phenotype also in later life. What we see is changes in the expression pattern, we see changes in epigenetic marks, and that means that the epigenetic mechanisms are here a kind of molecular memory of the adverse conditions during prenatal life. Major factor on this phenomenon of nutritional programming are, of course, the diets and the nutrients themselves, the genetic background of the organisms, and the timing of the nutritional supply. There are now quite a number of studies in farm animals and rodent models that have highlighted the effect of different diets on phenotype, transcriptome, proteome, metabolome, and also on the epigenome. In particular, protein, carbohydrate, and fat contents of diets were varied. The times before, during, and after pregnancy at which the different diets were used is also variable in the different studies, and indicated by the bars here on the plot. We fed different protein contents and different supplementation of components on the median metabolism in pigs throughout the whole pregnancy, indicated by the red and green bars here on the slide. The time of dietary shifts during pregnancy is an important factor. For this, we have to look at how DNA methylation are passed on through generations. Here we see the course of the methylation degree starting with the gametes. After fertilization in the zygote, the maternal and the paternal methylation patterns are deleted from the gametes, which is shown here, which represent the F0. And then there's a re-establishment of a methylation pattern in early embryonic stages, which is started here. So in this way, the methylation pattern is re-established in the somatic cells of the offspring, which is the black bar. And the offspring represents the F1 in this scheme. In prenatal development, also the primordial germ cells already are established in the fetuses. And these primordial germ cells represent already the F2. And also these primordial germ cells undergo a reprogramming of the methylation profiles. This means if you want to show transgenerational inheritance of effects of gestational diets, you have to look for the F3 generation, because only the F3 generation was not directly exposed to the nutritional shifts that were placed during the gestation. So the timing of the feeding during gestation is important. If you do it just to mid-gestation, you might not affect the methylation patterns of the primordial germ cells, so of the F2. If you do it all over the gestation, then you definitively have the F2 exposed to the different environmental conditions. Actually, what we did is we looked for fetal programming by using gestation diets with high and low proteins supply compared to the adequate maternal supply. We did this over the whole gestation and took a sample at late embryonic stages and at birthday. Animals were then kept with cross-fostering and with standard diets postnatally. And we had 2 other sampling points at 20 days of age and 188 days of age. The impact of the gestation diets with high and low protein content on gene expression was analyzed by us, and we also looked for the involvement of epigenetic mechanisms of DNA methylation in this study. What we did is we did comparison of the adequate protein supply diet to the high protein supply diet and also to the low protein supply diet in separate analyses. And here in this graph, on the vertical lines, you will find the number of differentially expressed genes between the 2 dietary groups at a certain developmental stage. And in the horizontal lines, you find some differentially expressed genes between the developmental stages within the diet group. And we especially looked then for those genes that are common in these timely shifts between the time points in both diets. So genes that are whose regulation are really due just to development. And we looked for those genes that were specifically regulated between time points in either of the dietary groups. So we did this for the high protein and for the low protein group. With regard to phenotype, I just want to mention that we found differences in fetal body weight and birth weight, but not on adult body weight. There were shifts in some metabolic traits, especially with regard to amino acid metabolism. So, of course, we did a functional annotation study of the differentially methylated gene, and just exemplarily, I would like to show here that you see that energy metabolism genes are affected at different comparisons. We found differences in steroid biosynthesis, in lipid metabolism, and in mitochondrial metabolism, just exemplarily. And to sum up the whole study, we can state that we have a hierarchy of effects on the transcript levels. With tissue, we compared liver and muscle, having a higher impact than developmental stage, and lowest impact comes from the gestational diet. We found that muscle is less resistant to nutritional modulation than liver. And interestingly, though there were some overlapping pathways affected, we found no gatekeeper pathways or genes. So that means there is not one pathway that is affected in both tissues and by both directions of dietary shifts. Interestingly, we found differential expressions of the DNA methyltransferases, so giving an indicating that epigenetic marks that DNA methylation plays a role here. And in fact, we found differential DNA methylation in a number of genes with implications on the metabolism. And as you can see by the upper right graph, there are significant differences of methylation of a number of CPGs within the promoter of one of these candidate genes. We did another fetal programming experiment where we applied a diet that varied in the content of components of the methionine cycle here. You see that methionine here is transferred to SAM, and SAM is the molecule that it used to transfer methyl groups to DNA, so which is involved in DNA methylation, and all the red marked components of this cycle were changed in our diets. And we fed these diets to sows of two breeds, Pietra and Landrace. We collected offspring at 4 different stages. So we had a 2x4x2 experimental design. Just as an example, we found an impact of the diets on the body weight, for example, in both breeds, left the DL and right the Pietra breed, where the methionine supplemented diets resulted in lower body weights. Interestingly, colleagues, the group of Dr. Braunschweig in Switzerland fed the same diets to boars. And if you do such fetal programming or nutritional programming animals experiments with boars, then you only need to focus on the F2 in order to show transgenerational inheritance. And actually what they found is that the F2 of the differentially fed boars differed with respect to back fat percentage, for example. They found in liver and muscle differential expression of genes related to lipid metabolism and other metabolic pathways. And interestingly, they found a significant difference in DNA methylation in the IYD gene at a p-value of 0.08. So this is an indication of transgenerational inheritance of differential methylation in a gene related to the thyroid functions, so with impact on metabolism. Actually, what we found is that there were some 400 to 2,000 differentially methylated sites, depending on stage, breed, and diet in this experiment. And of course, we looked for the nearest genes of these differentially methylated sites and found a number of genes there. And we looked for their functions, and affected pathways were the epithelial mesenchymal transition, the WNT signaling, adipogenesis, methionine metabolism, interestingly, and also changes of expression in DNA methyltransferases were shown. And we looked for the differential methylation of the offspring in this experiment, and we found significant correlation between methylation and expression of gene in the expected direction, so usually negative correlations for a number of genes. And what we specifically found that there are differences, of course, also between the breeds. You see here an example for a locus where we have, with a control diet, methylation of 55 and 67% in the DL and the Pietra breeds respectively. And the methionine-enriched diet led to a strong increase of methylation in the Pietra breed to 95%, but not in the landrace, in the DL breed. So there is a differential methylation between the breeds in response to the diets. What we can also show in this study is that differential methylation mostly takes place in regulatory sites and in transcription factor binding sites, as seen here with a view in the ATM promoter region, where you see largest differences depending on diet, always at those sites where we have transcription factor binding sites. What we found is that when we do cluster analysis on offspring of this experiment, we find some clustering due to stage, due to breed, but only a slight impact, obviously, of the maternal diets. So that means the impact of the diet is much lower than stage and breed, of course, and it is really difficult to find out which pathways are those that are most affected. And of course, it also makes it difficult to derive here whether there is any option to use the different diet to produce offspring with a certain phenotype. But I would like to finally mention the last experiment that we are running at the moment. Here we look for different phosphorus supply, and we fed mothers during gestation with 3 different levels of phosphorus. We had the offspring assigned to their mothers during lactation period, where the mother still received different diets with regard to phosphorus content, and then in the grower and in the fattener phase, we build up 3 dietary groups within each of the maternal groups. And what we could find is here that the serum P of offspring at low P supply is highest in those offspring that were derived from the mothers that received the low phosphorus diet. So that means piglets from mothers with low P supply use phosphorus best. And it is an indication that it might be possible to condition animals for low P supply. Yeah, you see that it is obviously a very complex story to derive the rules by which maternal diets might affect the phenotype of the offspring, and of course also the expression and the methylation patterns of the offspring. And it needs a lot of accumulated data from many differences until we will be able to derive rules and to make use of these rules. With this, I would like to summarize, and so what we heard in this presentation is that epigenetic marks, of course, determine the transcriptional potential of the cell. Epigenetic modifications provide, therefore, an additional level of variation that explains the phenotype, and knowledge of the epigenetic mechanisms and rules of origins and their inheritance will help to improve prediction of phenotypes, which will be useful, of course, also in breeding and especially in animal husbandry and management. Epigenetics does add to uncover the missing heritability and particularly to clarify missing causality. This is a very important point also with regard to genomic selection and the use of markers in animal breeding, because causal markers are, of course, the best one. Epigenetic modification mediate between the genome and the environmental factors, and these epigenetic modifications, in fact, represent the molecular memory of the nutritional programming phenomenon. Epigenetic studies can provide biomarkers for management, and I think if we really have collected enough knowledge on how certain diets and nutrients or other treatments may affect epigenetic marks and the expression patterns of animals, we might be able to develop certain phenotypes. And this would be possible from the generation 0 to the generation 1 that will be used in production. And for this gene, where we produce animals with certain responsiveness to environmental factors, it is not necessary to look for transgenerational inheritance of the epigenetic marks. With this, I would like to conclude by thanking my collaborators and all the founders of this study, and by thanking you for your attention. Thank you and goodbye.